Welcome to today's class. In previous classes, we have studied cleavage, blastulation and implantation, and today we will discuss the process of gastrulation. So what is gastrulation? Gastrulation is formation of gastrula or transformation of blastocyst into gastrula. Transformation of blastocyst into gastrula is called as gastrulation. So gastrulation begins on about 8th day after fertilization. 8th day after fertilization. So gastrulation begins on about 8th day after fertilization. So these are the diagrams. See, this is endometrium of uterus and this is morula stage. This is uterine cavity and this is morula. Morula enter in the uterus on about 5th day of fertilization. So this is morula in which cells are compactly arranged, and this is outer layer of cells visible in the morula. Then this after entry in the uterine cavity, morula absorb glycogen rich uterine fluid, that is called as uterine milk, and in the morula a fluid filled cavity is developed, and this cavity is called as blastocyst cavity. This cavity is called as blastocyst cavity or blastocele. And in blastocyst there are this is inner cell mass, or it is also called as embryoblast cell. This is also called as embryoblast cell, or it is inner cell mass. It is inner cell mass. And the layer, this outer layer of cells, it is called as trophoblast. This is called as trophoblast. So this is blastula or blastocyst. This stage is blastocyst. So on about 6th day after fertilization, blastocyst start adhering to the uterine endometrium. So blastocyst adhere to uterine endometrium, and on day 7 after fertilization, implantation start. So see here, implantation start. This is trophoblast, inner cell mass, and trophoblast cells form chorionic villi. So these are chorionic villi. And chorionic villi secretes lytic enzyme which dissolve or breaks the endometrium layer of uterine wall, and see, implantation begins as the trophoblast invade in the uterine endometrium. So this chorionic villi penetrate inside the endometrium. So this is day 9th of fertilization. So on about day 9th of fertilization, implantation continue and gastrulation start. So see, this is these cells are called as inner cell mass, but here cells are arranged in 2 layers, and this is the gastrulation. So this gastrulation we will discuss here, gastrulation. So implantation continue, and this inner cell mass forms bilaminar disc. So this is bilaminar disc. So we start gastrulation. So gastrulation, that is formation of gastrula from blastocyst, is called as gastrulation. So this is blastocyst, and this is the process of gastrulation start here. So conversion of blastocyst or transformation of blastocyst into gastrula is called as gastrulation, and gastrulation begins on about 8th day after fertilization. So during gastrulation, rate of cleavage or rate of cell division slowing down, but there is 2 important events takes place during gastrulation, and these 2 important events are Number 1, differentiation of cells. Differentiation of cells. And second event is morphogenic movement. Morphogenic movement. Differentiation of cells. So in the inner cell mass, cells are compactly arranged, and from the cells of inner cell mass, 3 primary germ layers are formed, and these 3 primary germ layers are ectoderm, mesoderm, and endoderm. And this is called as differentiation of cells. Differentiation of cells means 3 primary germ layers are formed from the inner cell mass, and this is called as differentiation of cells. Second, morphogenic movement. During the formation of these 3 germ layers from the inner cell mass, movement of cells takes place, and this movement of cell is called as morphogenic movement. So we will discuss gastrulation with the help of some diagrams. So gastrulation is formation of gastrula or transformation of blastocyst into gastrula. Gastrulation is actually the formation of 3 primary germ layers, that is ectoderm, mesoderm, and endoderm. So this is first diagram. In this diagram, so this is trophoblast. This is trophoblast layer. This layer is called as trophoblast, and cells are called as trophoblast cells. So trophoblast cells or trophoblast layer, and this is inner cell mass. This is inner cell mass. In inner cell mass, cells are compactly arranged. These are nuclei. So trophoblast, this is trophoblast, inner cell mass, and this is blastocyst cavity. Blastocyst cavity. This is blastocyst cavity or blastocele, which is filled with glycogen-rich uterine fluid or uterine milk. And the cells of trophoblast which are in contact with inner cell mass is called as cells of Rauber. So see, these layer I will draw with green pen. So these cells are called as cells of Rauber. So cells of trophoblast which are in contact with inner cell mass is called as cells of Rauber. So these are cells of Rauber. Cells of Rauber means the cells of trophoblast which are in contact with inner cell mass. So out of all the cells in trophoblast, only these few cells are in contact with inner cell mass, and these cells are called as cells of Rauber. So this is blastocyst. So how this blastocyst convert into gastrula step by step. So this is stage 1 or this is step 1. Cells of inner cell mass which are present on the free end. So inner cell mass, here cluster of cell, it is a cluster of cells, but see these cells which I mark, these cells are on the free end of inner cell mass. So cells which are present on free end of inner cell mass, so see these cells are present on free end of inner cell mass. So these cells are not present on the free end of inner cell mass. Only these cells are present on free end of inner cell mass. These cells become flattened and differentiate from inner cell mass, and they form a layer below the inner cell mass, and this is called as hypoblast. This is called as hypoblast. So cells which are present on the free end of inner cell mass become flattened and differentiate and arrange in the blastocell cavity, and this is called as hypoblast. This is called as hypoblast, and this is the endodermal cells. These are the endodermal cells which differentiate first from the inner cell mass. So this is hypoblast. Then second step, diagram number 2. So this is also, so trophoblast layer, cells which are present in trophoblast layer are called as trophoblast cells. So trophoblast cells will form placenta, and inner cell mass will result into embryo proper. So all parts of embryo will produce from this inner cell mass, and from trophoblast placenta will develop. Then cells, remaining cells of inner cell mass become columnar and arrange like this, and arrange in this manner. So, and this is below this hypoblast. So this is called as hypoblast, and hypoblast divide and redivide to form a sac in the blastocell cavity or in the blastocyst cavity inside trophoblast, and this is called as yolk sac. This is called as yolk sac. And the cells which become cells of the inner cell mass which become columnar and arrange like these are called as epiblast. This is epiblast, and this is hypoblast. This is hypoblast. So in the first step, cells which are present on the free surface of inner cell mass become flattened and differentiate and arrange within the blastocell cavity, and they form hypoblast. So these cells are endodermal cells which are which differentiated from the inner cell mass, and then all the cells which are present in the inner cell mass become columnar and arrange like this, like a plate, like a disc, and this it is called as epiblast. Hypoblast cell divide and redivide and form a sac inside the trophoblast, and this sac is called as yolk sac. So this is epiblast, this is hypoblast, and this is yolk sac. Next step, this is second step of the gastrulation. So in the third step of gastrulation, see, this is the third diagram in the gastrulation. So these are trophoblast cells These are trophoblast cells which are present on the periphery of the gastrula. So these are trophoblast cells. Now very important event takes place. Hypoblast cells. These are hypoblast cells. These are hypoblast cells, and this is, so sorry, I have to use black pen only. This is hypoblast, which form a cavity inside they arrange inside the trophoblast and form a sac-like structure, and this is called as yolk sac. So again, this is yolk sac. So this is yolk sac, epiblast. This is epiblast. It is hypoblast. Epiblast form from the columnar cells. These are cells which are present in the epiblast are columnar cells, but the cells which are present in the hypoblast are cuboidal. These are cuboidal cells, cube-like cells, and cells which are present in the hypoblast, they are columnar, like tall like a column. So these cells are columnar cells, and these cells are cuboidal cells. Now, cells of robber. So see, these are cells of robber. Cells of robber. Cells of robber degenerate. Cells of robber. So see, these are cells of robber. These are the cells of trophoblast which are in contact with inner cell mass. So we already discussed cells of robber. So cells of robber degenerate. Cells of robber degenerate. Cells of robber degenerate, and here there is a formation of cavity takes place. Here this is the cavity which is formed between these epiblast and trophoblast cells. So this cavity is called as amniotic cavity. This cavity is called as amniotic cavity. The cavity is called as amniotic cavity, and here cells of robber degenerate, but the trophoblast cells divide and redivide and form here a layer, and this layer of cell is called as amniogenic cell. So this is epiblast, this is epiblast, this is amniotic cavity, this is amniotic cavity, and see these cells which are present on the roof of the amniotic cavity, these cells are called as amniogenic cells. These cells are called as amniogenic cells. So these cells are called as amniogenic cells. These are amniogenic cells, and the structure which is formed by epiblast and hypoblast is called as bilaminar. It is called as bilaminar disc. It is called as bilaminar disc. So this is called as bilaminar disc. Bilaminar disc is formed from epiblast and hypoblast. So amniogenic cells, these are amniogenic cells which are present. Amniogenic cells are present at the roof of amniotic cavity. So this is the amniotic cavity. Amniogenic cells are present on the roof of amniotic cavity, and epiblast cells are present at the floor of amniotic cavity. So at the floor of amniotic cavity, epiblast cells are present, and at the roof of amniotic cavity, amniogenic cells are present, and amniogenic cells form extra embryonic membrane, and that is called as amnion. Amniogenic cells will form extra embryonic membrane, and that is called as amnion, which is formed from this amniogenic cells. So this is first step in the formation of gastrula or in the process of gastrulation. So inner cell mass, cells which are present on the free surface of inner cell mass divide and become flattened and differentiated and form a layer inside the blastocyst cavity, and this is called as hypoblast. So hypoblast are the endodermal cells which differentiate first from the inner cell mass. Then remaining these hypoblast cells divide and redivide to form a sac-like structure inside the trophoblast, and this is called as yolk sac. Then remaining epiblast cell become columnar and form a disc-like structure, and the layer of this columnar cells is called as epiblast. It is called as epiblast. Then cells of robber degenerate, and some trophoblast cell divide and redivide to form to take the place of cells of robber. In this stage, see, this is epiblast, this is hypoblast. Epiblast and hypoblast together form a bilaminar disc. So this is bilaminar disc. A small space or small cavity is developed between this epiblast cell and trophoblast cell. This cavity is called as amniotic cavity. Roof of amniotic cavity is lined by some trophoblast cells which are called as amniogenic cells. And these amniogenic cells will produce extra embryonic membrane, that is amnion. Epiblast and hypoblast together form bilaminar disc. So this is called as bilaminar disc. Then at the roof of amniotic cavity, amniogenic cells are present, and at the floor of amniotic cavity, epiblast cells are present, or epiblast layer is present. Next event is formation of trilaminar disc. So this is bilaminar disc, and after this formation of trilaminar disc will takes place. Actual gastrulation occurs about 15 days after fertilization, in which transformation of bilaminar disc 15 days after fertilization. 15 days after fertilization, bilaminar embryonal disc transformed into trilaminar embryonal disc. And this is actual gastrulation. So actual gastrulation occurs about 15 days after fertilization, and in which bilaminar embryonal disc transform into trilaminar embryonal disc, which takes place by division, rearrangement and migration of cells of epiblast. Cells of epiblast. So cells which are present in the epiblast divide, rearrange and migrate, and then bilaminar embryonal disc will transform into tribilaminar embryonal disc, and it takes place by formation of, it begins with formation of primitive streak and primitive groove. Formation of primitive streak and primitive groove. So primitive groove is formed on the surface of epiblast. So see how formation of primitive groove takes place. So this is bilaminar embryonal disc. This is bilaminar embryonal disc. These are epiblast cells. These are epiblast cells. Epiblast. So it is a bilaminar embryonal disc, and this is epiblast. The cells are epiblast cells. So these are epiblast cells, and below this epiblast cells, this hypoblast. This is hypoblast. This is bilaminar embryonal disc, and this bilaminar embryonal disc will transform into trilaminar embryonal disc on about after 15 days after fertilization. So formation of primitive streak and primitive groove takes place. Primitive groove form on the surface of embryonal disc, on the surface of epiblast. It is a shallow groove which extend from posterior to anterior, and see, this is primitive groove. This is primitive groove which is formed on the surface. And now the cells of epiblast divide and redivide. Cells of epiblast divide and redivide, and the cells means division takes place. Cells of epiblast divide and redivide, and then the cells move. See how movement takes place, how migration of cells takes place. The cells of epiblast divide and redivide, and then the cells migrate, and means move, and cells arrange here. So cells arrange here. These cells which I am showing by green color, these cells are derived from, these cells are originate from epiblast, and this layer is called as endoderm. So this layer is endoderm. So here this is endoderm. So this layer is endoderm, and due to formation of this embryonic endoderm, this is embryonic endoderm. So formation of this embryonic endoderm push this hypoblast layer and arrange here. So this is endoderm. Then again the cells of epiblast divide and redivide, and then some cells differentiate and arrange between epiblast between here epiblast and endoderm. And this layer, these are mesodermal cell. So this is mesoderm. So this layer is mesoderm. So first layer is endoderm. First layer which differentiated is endoderm. Second layer which is arranged between endoderm and epiblast, it is mesoderm. This is mesoderm, and remaining cells in the epiblast, they form, now this epiblast will form ectoderm. This is ectoderm. So see, this is trilaminar embryonal disc. So trilaminar embryonal disc formation takes place on about 15 days after fertilization. And how this bilaminar disc convert into trilaminar disc? So division, rearrangement and migration of cells of epiblast takes place, and it takes place by the formation of primitive groove and primitive streak. And primitive groove is a shallow groove which is formed on the surface of epiblast, and then the cells of epiblast divide and migrate. They migrate and they arrange above the hypoblast, and they push hypoblast layer, hypoblast cells, and they form a new layer, that is endoderm. So endoderm is the first layer. It is first germ layer which is formed. Then again cells divide from the epiblast, and that cells arrange between endoderm and epiblast, and then this second layer which is formed is mesoderm, and then from the epiblast ectoderm is formed. So in this way bilaminar embryonal disc transform into trilaminar embryonal disc, and process of gastrulation is completed.